about 6.5, neurons and synapses. Just a reminder before we start, you need to be taking notes on this, and then you are going to upload them as a PDF or as a Word document to ManageBack to get that stamp or 10 points in Plus Portal. So let's get started. All right, so when we're talking about the nervous system, for our purposes, we're going to focus on mostly how messages move through our body, which is part of the nervous system. So our essential question is, what are ways that signals get transmitted throughout the body? So think about, first of all, what might cause a signal to need to be transmitted through your body. I know we think of sometimes pain as a reflex that would need to signal something to our brain to make us try to run or to pull our hand away from something that's hot. But also think of it in a very simple way, like you see light when you first open your eyes, or when you see an image on TV. You're needing to send a signal to your brain to make sense of it. So it's not always a movement that needs to come from it. It could be just an action that's occurring within your brain. All right, so let's get started. How do they actually get transmitted throughout your body? Okay, so some key terms you need to know first about the nervous system is there's the central nervous system and the peripheral nervous system. The central nervous system is made up of your brain and spinal cord. So here in this picture you can see the brain, and the spinal cord here. The peripheral nervous system is going to be made up of your nerves, which are going to communicate with your motor and sensory signals between your central nervous system. So all of your nerves are going to be sending messages to your spinal cord and to your brain to then create an effect or an output, an action that needs to be taken. So you have the central nervous system, which is the core, brain and spine, and your peripheral nervous system, which would be your nerves. That sense a signal and then do an action. So just so you understand how fast these signals happen and that you can be thankful and grateful you don't have to think about it all the time, that it's a very automatic system, they can travel up to 268 miles per hour, which I believe is about 430 kilometers per hour. So super fast signals. So what we're going to focus on the most in this lecture is looking at the neuron. So this is a neuron here. If you're looking at this teal image here and then the yellow cord right here and then these little blue circles around it, this would be a neuron. Now this is also a neuron here that I'm circling, okay? And they're connected here to a muscle. So the nervous system is composed of neurons and they send rapid electrical impulses throughout your body. So if you were to flick your finger, that's going to be an impulse that is going to then need to be sent to your brain to make sense of what it just felt. Is it pain? Is it not anything to worry about? So this is your typical motor neuron here. It's a cell, so don't get that confused. It does look a little different. It's not circular like you would think of an animal cell to be, but every part of its structure has a function. So this is the nucleus, and it has all the other organelles inside of it that we've talked about before, like mitochondria, Golgi apparatus, lysosomes. Those are inside this whole cell, even though it's a funky shape. So here we would call the main cell body, and this is going to be the nucleus. These little arm-like structures or finger-like structures, we would say, are the dendrites, and then this long portion is the axon. Okay? In this picture, you can also see myelin sheets, which we talk about in your lab, and I'll get to more in a second, that are basically surrounding the axon. These portions here in the middle and these gaps are called the nodes of Ranvier. And basically, an impulse is going to be sensed by these dendrites and then sent down your axon to a muscle to make it move, potentially. But it had to travel all the way through a bunch of different neurons. So this neuron is going to send the impulse from the end here to this neuron here. So this is the anatomy of a neuron. Oh, and here, just to show you, this gap right here is called a synapse, which is going to be a way that the message has to be sent chemically to the other neuron. And we're going to talk about that more in a second, but here is the definition. The synapse is the junctions between two neurons. Okay, so we call this the reflex arc because you have to first feel something or sense something. Maybe this receptor is going to be in your eye. In this case, it's in a skin. So a sensory neuron, which is part of your peripheral nervous system, is going to be sending or relaying a message to your core of your body, to your spinal cord, up to your brain, and then it's going to send a message back to a motor neuron, which is then going to create an effect. So some sort of potential muscle movement maybe. So we call that the reflex arc. So a nerve impulse. A nerve impulse is in that lab that you guys are doing is that wave of energy that moves down the neuron. Okay? So the nerve impulse is conducted along a neuron. So here in this picture you can see they're calling it an electrical signal. And the reason the reason this happens is there's a change in concentrations of sodium and potassium ions across a membrane. And so we're going to look at that more carefully in an image. But without that sodium-potassium exchange, or depolarization, and then again a repolarization, this would not happen. So there's two terms we use when we're talking about the movement of an impulse. There's the resting potential, which is what you're at when you don't need to signal your body. So it's an electrical potential, so that the potential is there, but nothing's happening yet. So an electrical potential across a cell membrane when not propagating an impulse. An action potential is when you are actually going to charge or send an impulse. So it's the reversal, then the restoration of an electrical impulse. So basically, it's going to it has to depolarize, and then it needs to reverse that. It needs to repolarize it and bring it back to its resting potential. And this happens as it passes along this axon. This graph is showing this here, but it'll make more sense more in the next slide. But just so you can see, it says membrane potential on the y-axis at a -70. It increases, becomes more positive in the depolarization, and we call this point the action potential. And then it repolarizes and comes back down to that resting potential, which is right here. Okay, so a resting potential, like we said, is the electrical potential across the plasma membrane of a cell that is not conducting an impulse or not propagating an impulse. So the resting potential is maintained by active transport across something called an antiport. An antiport is, I'm going to jump ahead a few slides really fast, is something like this right here that I'm circling. It is a cell that it stays open, and it lets two different types of ions diffuse between it. So it's slowly releasing out some potassium and letting in some sodium to keep it at a balance, to keep it at a resting potential. So let me slide back here. Okay, so what's occurring here inside the neuron is the neuron is more negative on the inside, and the outside is more positive. So during the process of an action potential, there's going to be movement of these sodium ions, and they're going to be coming inside, and then there's going to be movement of potassium ions going outside. But to get it back to a resting potential, which is what they're showing you here, the sodium that went inside needs to now move out, making this negative again, and the potassium ions need to move back inside, which is gonna seem a little weird because you're gonna notice the potassium ions have positive charges, but there's less of them inside, so that's gonna make it still be more negative than the outside. This is gonna make the neuron, remember, anything that has a negative charge on one side and a positive charge on the other, like a battery, is going to be considered polar. So that's why you keep hearing me use the words depolarization and repolarization. So in this picture you're seeing it get repolarized and ending at resting potential. Okay, action potential. So these are the two steps to action potential. So notice here that it's negative first on the inside and positive on the outside. But during an action potential, what's going to occur first is there's going to be sodium channels that are built into this plasma membrane here, into this phospholipid membrane, and it's going to open those channels and it's going to send inside a bunch of sodium. So notice it becomes positive now because now there's potassium inside that's positive and a bunch of sodium ions that are positive. So it has become depolarized because now it is more positive here on the inside. So that's depolarization. Then, after there is enough sodium on the inside, it triggers repolarization and the action potential has occurred. It's going to trigger repolarization. This is when sodium channels are going to open and they're going to send the potassium to the outside. And then this is going to create the negative internal charge again, and we call that repolarization. So you've heard me maybe use the word threshold potential, and this is a critical level that has to happen in all animals in order for any type of action potential to happen. So what that means is there has to be a certain amount of sodium that comes inside the neuron, and it has to hit that threshold or the action potential will not occur. These threshold potentials are necessary in order to regulate and propagate any type of signaling in both the central nervous system and the peripheral nervous system. So what is that number? What is that threshold? So if you look here at the picture, the resting potential is always negative 70. So the charge that's happening inside the neuron. So we're looking at the inside of the neuron with this graph. So here's number 1. You can see it's at a resting potential. This antiport channel is open, allowing some of the sodium ions to flow in and some of the potassium ions to flow out, or vice versa. When depolarization has been put into effect because some sort of sensory message needs to be sent to the brain, you're going to see sodium start to come inside. So here you're seeing sodium is coming inside the neuron, which is making it more positive in here. So that's why this is starting to go up. It's becoming more positive. So notice on the number line, that would be going towards the positive side. So it's going to be becoming more positive. When it hits negative 55, it's always negative 50, negative 55-ish range. That's the threshold. And if it passes that point, the message or the action potential is going to happen. If it doesn't pass that point, there'll be a dip back down to resting potential. So since it has hit that point, once the action has been sent and the message or impulse has gone down the neuron, it has to repolarize itself. So it has to become more negative on the inside again. So that means if you look at this picture, potassium ions are going to start going out, and it's going to start evening out and balancing this back to a negative charge on the inside. So notice this part here, it tells you where it's negative on the inside and outside, then depolarization happened, it became more positive here, negative outside, and now repolarization is happening, so it's more negative here and positive outside, and now it's back at its happy place at resting potential. Notice this dip here. This dip is important because it takes a little while for the neuron to repolarize and get to resting potential, so no action potential can happen for at least 2 minutes, or notice this, milliseconds, before another one can happen. Milliseconds, not minutes. And sometimes even in the lab you saw, it might take up to 10 milliseconds before another action potential could potentially occur. Okay, to those myelin sheaths. So here is that picture of a neuron. This is one neuron, and this is myelin sheaths that are wrapped around the axon. What myelin sheaths do is they control the speed of the impulse going down the axon. It actually speeds them up. And it's insulating that axon. Oops. Because the myelin sheaths are there, it allows the axon, or it allows the impulse to do something called saltatory conduction. Basically what this is the impulse is jumping from node to node. No sodium and potassium can move in or out inside the myelin sheath insulation. It can only happen in these gap places where there's no myelin sheaths. Because of that, there's less distance for the impulse to travel, so it hops from node to node to node, making it basically be a shorter distance to send the message, and we call this saltatory conduction. So what is the purpose of the myelin sheath? Or why is the myelin sheath of surrounding nerve fibers important? It's because it makes the impulse go faster down the axon. All right, so we've talked about how it's moved through the main cell body, but now we need to talk about how it gets from neuron to a different neuron between this gap. How does that message fly across this gap here in this picture? So this is the dendritic end, and this is the terminal end of one neuron, and this is the dendritic end, so the beginning of another neuron. And this is what we call the synaptic gap. So an electrical impulse now has to be converted to a chemical neurotransmitter. So think about hormones here, a chemical exchange. So you're going to see it go from something that seems very electrical as it's moving through the neuron to something that is more chemical. So we're going to talk about more how that happens. So here is another way of looking at it. Here is the end of that original. So the message is coming this direction. This is the end of the axon. And this is a dendrite, one of those finger-looking things on a new neuron. So what's going to happen here, or let's just look at the names of some of these key players. So we call this the presynaptic neuron, and this would be the postsynaptic neuron. In order for it to change from an electrical impulse to a chemical impulse, you need something called this voltage-gated calcium channel or calcium pump. So it's kind of like the sodium-potassium pump, except now we're using calcium. It is an active transport method, just like the sodium-potassium pump. So energy is needed, hence these mitochondria here, in order to make it happen. There are these vesicles that are carrying neurotransmitters, and these neurotransmitters are going to be the chemical message. So they are the key neurotransmitters, and they are going to float their way across. And notice their shape, because structure always determines function. So their shape is going to match with one of these neurotransmitter receptors, and when they attach, it's going to then trigger the sodium-potassium pump on this side to make the action potential continue down the next neuron. So this basically goes through all the steps I just talked about, so you could work your way through it and see what step 1 is, step 2, the calcium pump here is going to, so step 1, the neuron impulse comes down into the end of the axon. Step 2, this is depolarization, the last depolarization of this neuron, and it's going to be calcium moving in to cause a change in the voltage or the charge here. Energy is needed to make this happen, so it kicks off one of the phosphates, the ATP, and makes it adenosine diphosphate, and that causes this pump to move. This calcium then triggers the neurotransmitter vesicles to move towards the end of the membrane of this neuron, which then those neurotransmitters are going to diffuse across the synaptic gap into the postsynaptic receptors or neurotransmitter receptors. Once they bind, it's going to trigger the sodium channels to open, causing depolarization over here and an action potential to occur and move down the next neuron. After that has happened, the neurotransmitters that have been floating around here in the synaptic gap are going to be broken down by different enzymes and absorbed back into this axon here. And then the process repeats itself when it's needed. An example of a neurotransmitter, there's a bunch of them, but acetylcholine is a type of neurotransmitter, and it's found between neurons and muscle cells, and other places, but muscle cells is the main focus. This is its structure here, this is what it looks like. And so it's a chemical, remember, it's not an electrical charge, it's going to be like a hormone. If you break down the name, acetyl from respiration and choline from diet, so acetylcholine, it's going to travel across the synapses to bind to receptors, and then it'll be the enzyme acetylcholinesterase, remember enzymes always end in ase, will rapidly break it down. An acetylcholine in the synapses breaks it back down into choline and its acetate, and then it gets reabsorbed into that presynaptic neuron to be reused later. So this would be an example of a type of neurotransmitter. And this works through, you can see exactly what I just said with the stages of release, it binding, and then deactivation. So one way that we interact with our neurotransmitters are through drugs. And drugs can be used in positive ways for a body and negative ways, obviously. And remember that drugs, there's a certain tolerance level in all living things. So a certain type of drug that might help me may not help if it's in the environment for another animal, like a bird or a frog that might end up drinking it in a waterway that our drugs have been released into. And so it might affect their neurotransmitters differently than mine. So keep that in mind about all organisms are going to react to these drugs differently. So things that drugs can do to affect neurotransmitters. So what I'm talking about is happening in this area here. So it can block a receptor of a neurotransmitter. So that would mean that the receptors were filled in by a drug, a different hormone drug, so that the action can't take place. It can block release of a neurotransmitter, so meaning this won't happen here, where these drugs are the neurotransmitters released, so that'll make the action potential not happen in the next neuron. It can enhance release of a neurotransmitter, so it could make it go faster. It can enhance neurotransmissions by mimicking another neurotransmitter, so maybe your body's lacking in a neurotransmitter and it's giving you more of it, so that you can you can function appropriately. And then it can also block removal of a neurotransmitter. So these neurotransmitters here won't be able to be broken down by enzymes and reabsorbed. So we have drugs that can do all these things. Botox is an example of blocking a neurotransmitter. So it's making the muscle not move. There's a block here. And so that muscle cannot move at all, and that's why when people get Botox injections, it helps prevent wrinkles because the muscles won't move. Here's an example of a different, here's an example of a type of drug. It's an insecticide, and basically it's called a neonicotinoid, okay? And it falls into this family of this family of drugs. So a neonicotinoid is a synthetic chemical similar to nicotine. So what it does is it binds to the acetylcholine receptor irreversibly. So when it binds to that receptor, it leads to paralysis because basically the message can't be sent down the next neuron. And in insects, this can cause death because they can't move anymore. So neonicotinoids are very widely used because we thought they're safe for mammals. So what's the problem here of using them on insects? Well, what we've found is it is non-specific, meaning that it doesn't only affect the insect we care about, it can affect all insects. So this has been a big issue with killing helpful insects like bees, because they become paralyzed and can't move. So neonicotinoids are a big issue with having them in insecticides that farmers use and spray on their crops. So having brands that will now not use those has been very helpful in reviving some of these bee populations. So this is an example of a drug that can block our neurotransmitters from working effectively and moving an action potential through our body. Okay, so we're going to stop here at this point. So please make sure you took notes on that lecture and that you load them into ManageBack and that you make it a PDF or a Word document. I hope that was helpful. Let me know if you have any questions.